This lesson is about meiosis. Meiosis is the division of sex cells, sperm and egg. Sometimes they're called gametes. But let's remind ourselves of the cell cycle. So typically, cells spend most of their life in interphase. G1 is when they're growing, and then there's a checkpoint to see has the cell grown large enough before it can start the S phase. In the S phase, DNA is replicated, and the cell continues to grow in the G2 phase. There's another checkpoint again to make sure that the replication has gone well and that the cell is large enough. If so, it starts the process of mitosis, prophase, metaphase, anaphase, telophase. During mitosis, there's another checkpoint to make sure that the chromosomes have been equally distributed. Then the cell will divide into two new cells, cytokinesis. To understand meiosis, we need to understand a little bit more about chromosomes. The picture here is a diagram of a chromosome. A chromosome is just coiled up DNA, and this chromosome has a center point called the centromere. It's the centromere that will attach to spindle fibers and make sure that the chromosomes are equally divided or distributed to the two new cells. But during that S phase, when chromosomes are being replicated, what I have here is a picture of a duplicated chromosome. So this duplicated chromosome, we're going to see that in prophase. So the first time we'll be able to see clearly a duplicated chromosome like this. You'll notice that it still has the one centromere, but now we have these two parts that are genetically identical, and they're called chromatid. So we still count this as a single chromosome, but it's made up of two sister chromatid. Now that gets a little bit crazy when we're counting chromosomes. In the first picture, we can see that there's 12 chromosomes. In the second picture, there are also 12 chromosomes. What makes it interesting is in the second picture, we have 24 chromatid because all of those chromosomes have been duplicated and now there's 24 chromatid. And that's essentially what's going to be distributed to the new cells. So again, let's just review mitosis. We start with 3 chromosomes because mitosis is really just the division of the material in the nucleus. So I start with 3 chromosomes, and the question is, how many will I have in my new body cells? So this is prophase, the nucleus is starting to disappear, the chromosomes are clearly visible. And we enter metaphase, where the chromosomes are lined up in the middle. Anaphase, they start to move to opposite sides, and it's the spindle fibers that are attaching to these centromeres that are making sure the chromosomes are moving to opposite sides of the cell. And then we have telophase, where the nucleus starts to reform and the chromosomes are in opposite sides, and we're about to get 2 new cells through the process of cytokinesis. So that's where all the rest of the organelles in the cytoplasm are all going to be distributed evenly, and we have 2 viable cells. But notice that each cell has 3 chromosomes. We started with 3 chromosomes, we end with 3 chromosomes. That's the idea of mitosis. What about meiosis? Well, meiosis occurs in 2 phases, meiosis 1 and meiosis 2. Some of the things to look for when you're in meiosis 1 or observing meiosis 1 is look for pairs of chromosomes. Homologous chromosomes just means the chromosomes are the same size, same shape, have the same genes. And now we're going to look at these homologous pairs of chromosomes. It's the pairs of chromosomes that become visible in prophase, and they even cross over in prophase. We'll see what that is in a moment. In metaphase, those pairs of chromosomes are lined up. And in anaphase, those chromosomes are moving to opposite poles, but they're made of sister chromatid. And then in telophase, we see the sister chromatid pairs are in the newly forming nucleus. Here's a homologous chromosome. Notice that it's the same size. The 2 chromosomes are the same shape, same genes. And we're counting them as 2 chromosomes because of the centromere, or we're only counting the centromere. But they're chromatid and they're duplicated. All right, together they make a homologous chromosome. This is what crossing over looks like, where again we have the chromosomes are together in prophase, of meiosis 1, and you'll notice that they swap some genes. Now that's important because it does increase variation. And it is important to note that some genes are more likely than others to swap. It's only those parts of the chromosome that are far from the centromere, that are to the end of the chromosomes that get swapped. So it's not often pieces of chromosome close to the centromere that get swapped. So here's meiosis 1 again, and we're looking at these pairs of homologous chromosomes as they go through prophase, metaphase, anaphase, telophase. Meiosis 2, now we are looking at two cells that are dividing simultaneously because our end result is going to be 4 cells. So notice that we have the sister chromatid in each of the nuclei of these cells that are in prophase 2. There is no interphase 2, so it just starts with prophase 2. We go to metaphase 2, they line up in the middle, and then anaphase 2, they start to go to opposite sides, and you'll notice that one sister chromatid goes to one side, the other sister chromatid goes to the other side. And that's what's going to make those two new cells. Meiosis is the division of sperm cells and egg cells, sex cell division, gametes. In the case of sperm, you'll notice that when the cells divide, all the sperm cells are essentially the same size, and all 4 cells are going to survive. With the egg cells, it's different. The egg cells, the cytokinesis is different, so that more of the cytoplasm and organelles go to one than the other. And as a result, we have 3 very small egg cells that don't survive. They're called polar bodies, and we have the one large egg cell that does survive. Sometimes things go wrong in meiosis 2. One of the problems with meiosis is something called non-disjunction. Whenever the chromosomes aren't separating properly, and in this case, you'll notice that we wound up in telophase with too many chromosomes in one of the nuclei and too few in the other. Now, non-disjunction usually results in cells that don't survive, but occasionally they do, in which case we have all kinds of genetic disorders. For example, Down syndrome, Klinefelter syndrome, Turner syndrome are all examples of sex cells that either have too many or too few chromosomes. So I hope that helps you understand a little bit more about meiosis and the process of sex cell division.